Welcome back to Scientia Academiae. Today we are going to continue our O-Level Biology Review series. In this video, we'll be tackling questions from the May-June 2016 session, focusing on paper 21 and 22. Let's dive right in. We start with question 7, which carries 14 marks. Figure 7.1 explores genetic engineering, specifically the production of insulin. Let's break it down. Part A has 5 parts. Now let's look at them one by one. In Part A, Part 1, we need to describe how the location and organization of genetic material in human cells differs from that in bacterial cells. In human cells, genetic material is located within the nucleus, which is enclosed by a nuclear membrane. This DNA is organized into paired, linear chromosomes. Conversely, bacterial cells have their DNA located in the cytoplasm, organized as a single circular chromosome along with additional plasmids, and lack a nuclear membrane. Moving on to Part A, Part 2, we need to identify 2 structures the transformed plasmid must pass through to form a transformed bacterium. To form a transformed bacterium, the transformed plasmid must pass through 2 key structures in the bacterial cell. First, cell wall, and second, cell membrane. These are the primary barriers the plasmid must traverse to enter the bacterial cell and integrate into its internal machinery. For Part A, Part 3, we're looking at the type of reproduction in stage M. Use your knowledge of the process of cell division to explain why it is important that this type of reproduction occurs. In this stage, mitosis occurs. This type of reproduction is important because it produces genetically identical cells, ensuring that each cell, or clone, can perform the same function and produce the same product, such as insulin. In Part A, Part 4, name the condition in humans that is treated using insulin produced by the bacteria in stage N of figure 7.1. The condition treated with insulin from bacteria is diabetes. Finally, for Part A, Part 5, stage N of figure 7.1 may take place in a container similar to that used in the large-scale production of antibiotics. State the name of this type of container. The container used in the large-scale production of insulin is called a fermenter. Let's move on to Part B, which discusses genetic engineering in crop plants. We'll weigh the advantages and dangers. Potential advantages include increased crop yield and profitability, accelerate growth, and enable crops to thrive in extreme conditions. It offers more predictable results and the ability to transfer beneficial genes between species. This can lead to nutritionally enhanced, visually appealing, and disease-resistant crops. However, there are dangers to consider, which include potential genetic spread to wild species, high costs due to patents, and unknown health or environmental impacts. There could be unforeseen effects on other species and ecological balance. Next, let's explore question 8, that carries 11 marks. Let's start with the question. Figure 8.1 shows the leaves of the same plant during daylight and during the hours of darkness. In Part A, name a process that takes place in the plant in figure 8.1, A, only during daylight, and explain how the leaves help this process. We identify photosynthesis as the process occurring in daylight. This happens in daylight because leaves are adapted to capture sunlight. Their broad surfaces and chlorophyll absorb light, while stomata take in CO2. Transparent cuticles and thin leaves let light through, and veins supply water and remove products, supporting efficient gas exchange and light absorption. In Part B, the folding of the leaves at night, as seen in figure 8.1, B, is called a sleep movement. Suggest and explain the effect of these sleep movements on the loss of water from one of these plants. The effect is that it reduces transpiration. The sleep movements of leaves, which involve folding at night, help reduce transpiration by limiting water loss. When leaves fold, the stomata, mostly located on the lower surface, close or open less frequently, leading to a buildup of humidity around the leaf. This increased humidity reduces the diffusion gradient for water vapor, resulting in a lower evaporation rate. Additionally, the folding reduces the leaf's surface area exposed to the air and provides protection from wind, further decreasing water loss. In Part C, Figure 8.2 shows the uptake and loss of oxygen by a leaf during a 24-hour period. In Part C, Part 1 explain what is happening in the leaf between 1800 hours and 2000 hours. Between 1800 and 2000 hours, as light levels drop, photosynthesis in the leaf slows down, leading to reduced oxygen production. To conserve water and limit carbon dioxide intake, the stomata begin to close. In Part C, Part 2 explain what is happening in the leaf after 2200 hours. At 2200 hours, in darkness, or low light, the leaf stops photosynthesis and starts absorbing oxygen for respiration. It uses this oxygen to break down glucose for energy, producing carbon dioxide and water as byproducts. The leaf may also continue to absorb water from the soil to maintain cellular functions. The last question, question 9, carries 7 marks. Malaria is a disease caused by a parasite that is transmitted from one person to another by a vector. In Part A, name the vector of the parasite that causes malaria. The answer is that the vector for malaria is the Anopheles mosquito, which transmits the Plasmodium parasites causing the disease. Part B is divided into further 2 parts. Let's look at these parts one by one. Part B. Part 1 is about the spread of the vector may be controlled by using an insecticide. State 2 other ways of controlling the spread of the vector. Besides insecticides, we can control the spread of mosquitoes by: firstly, regularly draining swamps and preventing the stagnation of water can reduce the breeding habitats for disease vectors like mosquitoes. Secondly, applying a thin layer of oil on the surface of standing water can prevent vector larvae from breathing, thereby controlling their population. Part B. Part 2 states that resistance to the insecticide can appear in the vector population. Describe how the process of natural selection may bring about resistance of the vector to insecticide. Insecticide resistance in a vector population arises through natural selection. Initially, some vectors have resistant alleles. When an insecticide is used, susceptible vectors are killed, while those with the resistant alleles survive and reproduce. Over many generations, these resistant vectors become more common, leading to an evolutionary shift where resistance is prevalent in the population. And that's a wrap on today's review. We hope this breakdown helps you in your studies. Don't forget to like, subscribe, and hit the bell icon for more content. See you in the next video.